We are recording. Let's see. Case bolt. Yeah, case bolt, right? Case bolt. Case bolt is here. Computers, right? No, not. Mankins is here. Yep. Thompson, that's what I was thinking of. Thompson, just signed your paper. Thompson's here. Mathis is here. So far, that's all for people who are physically here. Reminder, if you're online, you have to send the words in to get the extra credit and to be counted as present. Less than a minute left. So I'll go ahead and do attendance again. Call out the people who are not here too. These are the people who are currently physically here and not currently physically here. Bangura is not here. Case Bolt is here. Tristan is not here. Covington Griggs is not here. DeVere is not here. Dow is not here. Hughes is not here. Lebdy is not here. Carla is not here. Mankins is here. Mathis is here. Rains is not here. Reed is not here. Elise is not here. Thompson is here. Yandui is not here. Elkins is not here. So again, that is the list of people who are currently not here, and it is 8 a.m. I'll wait till 8:01, and then we'll get started. Get things ready. Wow, we're waiting until 8:01. So, since on any given day, half of the class is either not here when we start, or they come in late, or they don't come at all. So because of that, I'm not actually going to start with announcements and reminders. I'm going to do that more towards the middle of the today's presentation, because I keep repeating myself and people keep making the same mistakes. So my assumption is it's only because they're either not here and seeing the same things I keep repeating at the beginning of every lecture, or they're coming in late. So again, they're not seeing the things that I had presented at the beginning of every lecture. Let me make sure we're recording. Oh yeah, we are recording. Double check that. Yes, we are recording. So again, well, I guess anyway, before we get started then, of course I'm preaching to the choir if you're here and listening, but if you find yourself running late, maybe just don't come. Use the link and join us online, that way you don't miss anything. Or if you were late, what you could do is watch the video, right? You're missing important stuff. So if you come in late, or excuse me, yeah, if you came in late or come in late, whatever, then you should watch the videos when I post them. If you don't know where they are, email me, I'll show you. Or you can read the announcements from the beginning of the semester. Someone's asked, oh my goodness, okay. Anyway, yes, so let's get started. Again, if you come in late, watch the videos. Also, make sure you let me know that you're late. So anyway, let's jump into this very quickly. We might finish this chapter. My goal is to finish a chapter today, but we might not finish it today. And if we don't finish it, whatever we cover today is what we're going to do. Whatever we don't finish, I'm just leaving it up to you to read on your own. I know that sounds horrible, but let me explain. This chapter we're about to cover is not on any exam. We're going to cover the basics of cells and really quickly, because all the important stuff, the most important stuff about cells that you're going to learn are going to be the next following chapters. Like, for example, after this chapter, you're going to learn about how cells use energy, and then you're going to learn how they convert energy from one form to another, things like respiration and photosynthesis, and you're going to learn how cells reproduce. So this chapter, as far as the exam is concerned, or exams are concerned, very unimportant, completely unimportant. As far as the semester is concerned, it's kind of unimportant because, again, all the stuff that you need to know about cells you're going to learn in the following chapters. So this is all just kind of a preview, some of which is going to help you in the next chapters, and the rest is going to be stuff you don't need to know at all. So anyway, I guess the first word for attendance, going back to what I was saying on Wednesday, because remember, it's not a word. I'm going to ask you a question. So the first attendance word, official attendance word, is you answering this question. Will anything from this chapter be on the exam? Is the first word for attendance. So it's a yes or no is the first word for attendance. Yes or no. Anyway, let's talk about how cells are studied. This is not going to help. This is, again, not going to be on the exam. And this isn't really going to help you later in the semester. But it's in the textbook. It's a 100-level course, so I'm testing out of the textbook. All right, the learning objectives of this section are, by the end, you should be able to describe the roles of cells and organisms, like what do they do. That's slightly important, but again, you're going to learn more about that later. You should be able to compare and contrast light microscopy and electron microscopy. Again, for us in this semester, very unimportant. I'm going to go through it so quickly. And then you should be able to summarize the cell theory. And once again, it is important that last bullet point is important in the grand scheme of things, but you're really going to learn more about it throughout the semester. So any questions about the learning objectives? It looks like I got my stuff flipped. Anyway, that's okay. Microscopy, very quickly, there's different types of microscopes, right? Here you have a light microscope. Here you have a dissecting microscope. Not pictured as an electron microscope. Again, I'm not going to ask you anything about those. If you want to for independent work, you can look those three things up and kind of compare and contrast them. You can get really deep into it, have a long paper, or you can barely scratch the surface and have a short paper. Which reminds me, independent work is now down to 13 points per page. 13 points per page for independent work. So, as time goes on, obviously, it's getting lower and lower. When we first started on day one, I said it's worth 10 points. And then I said, but to bribe you, for now, it's worth 20 points, right? It keeps getting lower and lower. So, turn in some papers. Help yourself.
When do those go into the grade book?
Oh, at the end, well,
Yeah.
I have to decide that. That's a great question. So the way the grade book works is like, let's say you've turned in 50 points worth of work already, which is great, right? Because we're not close to being done yet. But if I were to put those 50 points into the grade book, it's all mathematical.
Mm-hmm.
So the grade book just looks at that as 50 points. You've only turned in 50 points out of 100, which would then hurt your grade.
Okay.
So what I usually do is I'll not put anything into the grade book until you hit the full 100 points. Or I think usually after about midway, so after midterms, then I will put in what your grade is. And that will, so your overall grade, unless you have all the independent points, your overall grade will appear to have dropped at that point. But I want to do that. That way I don't want to do that at the end. Like you think you have oh man, I've got a B, I'm good to go. And then it's the end of the semester and I put those grades in and it drops you down to a C or something. So I don't want to surprise you with that at the end. So great question. And so yes, the grades will go in either when you hit 100 points, which one of your classmates is about to do that, or at midterms, after I turn in midterm grades. Because that's the last time I turn in grades until the end of the semester. All right, cell theory. Let's talk about this very quickly. Again, I'm not going to ask you any questions about this, but the unified cell theory states that all living things are composed of one or more cells. We already know that from chapter 1, which will be on the exam, right? That's one of those properties of life, that all living things are made of cells. The cell is the basic unit of life, kind of the same thing we're saying there, just in different ways. And that all new cells arise from existing cells, right? So other than the first cell that ever came into existence, all other cells came from those cells. I've got some documentaries. They're on that list of videos that you guys can watch for extra credit. If you don't know where that list is, let me know. If I think of it, I'll share those videos, at least at Facebook. I don't like to share a lot of small stuff on the announcements because I don't want to overwhelm you with emails. Anyway, Leeuwenhoek, he was the first to observe bacteria and protozoa. That might be an extra credit question. Oh, no, there we go. I'm sorry. This will probably be, I think this is an extra credit question. If there's going to be extra credit questions on the next exam, this one might be one of them. This is all in your textbook, by the way. Robert Hooke coined the term cell. So again, good independent work topics. You can talk, look into these two people, write a little bit about them. Write a lot about them, write a little about them, whatever you want. Anyway, both of these two scientists are credited for the discovery of microorganisms. So any questions about that? It's important for biology, but this actual biology, but it's unimportant as far as understanding the information I'm going to give you this semester. All right, nice and easy. We'll move forward then. We'll talk about comparing prokaryotic and eukaryotic cells. The learning objectives of this section are, you should be able to name examples of prokaryotic and eukaryotic organisms. For example, I'm a human, you're a human, we are eukaryotic organisms. I ate a banana for breakfast today. That's a eukaryotic organism. I have bacteria in my stomach. Bacteria are prokaryotic organisms. Of course, I'll give you some more examples a little bit later. You should be able to compare and contrast prokaryotic cells and eukaryotic cells. Maybe there'll be a test question on that, but it'll be very simple, and you'll see. And like I just said, we are eukaryotic cells, as for one example, and bacteria are prokaryotic. So if you can remember those two examples, then all this information I'm about to give you should be pretty easy to remember which is which. And you should be able to describe the relative size of different kinds of cells. So here we go. Here's some things that all cells have that you're going to learn more about in the upcoming chapters. All cells have plasma membranes, whether they're eukaryotic or prokaryotic. They all have plasma membranes. What is a plasma membrane? Is the outer covering that separates the interior from its surrounding. So last chapter, when we were talking about biological molecules, when we were talking about lipids, there was different types of lipids. One of the types of lipids I talked about was a phospholipid bilayer, phospholipid. And that's what most of the plasma membrane is made up of, is those phospholipids. And again, you're going to learn more about that a lot in the next chapter. So now it's just more like, hey, let me introduce you to this concept. All cells have cytoplasm. That's basically the inside stuff. It's a jelly-like region where all the other cellular components are found. So you've got the plasma membrane, which is the outside, and then you have the inside, which is the cytoplasm, and then all the stuff that's inside the cytoplasm. All cells have DNA, as we talked about in the last chapter. You do need to know about DNA because we talked about it in the biological molecules discussion. And like I mentioned then, there'll be an entire chapter on DNA. So for now, you only need to know a little bit about DNA. Later, you'll need to know a lot about DNA. Ribosomes. All cells have ribosomes. What are ribosomes? Ribosomes are the things that make proteins. And again, we'll have an entire chapter where we talk about how we go from DNA to RNA to protein. At that point, I'm really going to introduce you to ribosomes. And at that point, you're going to need to know about it. For now, it's just an introduction. Am I hearing dogs?
Yeah, there's some.
Thank God I'm not losing my mind. Well, I might be, but so that's not, I'm not missing that. Anyway, like I said, all of those will be discussed later. Any questions about that slide? Okay, so there's two types of, again, we're going to compare prokaryotic and eukaryotic cells. The first one we're going to talk about is prokaryotic cells. These are the most basic cells. And there's two different types. There's bacteria and archaea. Again, I'm probably not going to ask you any of that later. And then, of course, there's eukaryotic cells, which we're going to talk about later. So again, prokaryotic cells are broken down to bacteria or archaea. As far as we're concerned, those two things are basically the same. Right? So in a sense, for this 100 level non-biology major class, mostly these three words can be interchanged. Right? Prokaryotic cells, There's either going to be our bacteria cells or archaea cells. As far as we're concerned, they're pretty much the same. Now, if you're really digging deep into it, there's actually a pretty big difference between archaea and bacteria. And you can look it up for independent work if you want. What is the difference? That could be a quick 2-sentence paper. Like I looked it up, and according to this source, the difference between archaea and bacteria is this. And you could probably summarize it in 1 sentence. Or then you could probably dig a little bit deeper if you want. Give me some examples of this, some examples of that. So anyway, prokaryotic cells. They're very simple. They're the most simple cells, actually. These are single-celled organisms. So all prokaryotes are single-celled organisms. Let me make sure this is clear. This will be a little bit more later. All prokaryotes are single-celled organisms, which is not the same as saying all single-celled organisms are prokaryotes, right? We'll talk about that later. Sort of like saying all pickup trucks are automobiles, but not automobiles are pickup trucks. Same thing here, right? So all prokaryotic cells are single-celled organisms, but not all single-celled organisms are prokaryotic. They don't have a nucleus. As a matter of fact, they don't have any membrane-bound organelles. And we'll talk a little bit more about organelles later. But basically, it's the things inside of the cells that do work. They do things. And again, the important ones we're going to talk about a lot in the next chapters after this. Again, like we said earlier, all cells have DNA, including prokaryotic cells. This is, in this case, the DNA is found in the central part of the cell, which is the darkened region called the nucleoid. So again, information you're not going to need. This is a 100 level course, so I'm teaching out of the textbook, and I'm flying through it for this reason. So I'm not going to ask you any of these questions. This is an introduction to stuff you're going to learn later. And to be honest, we're barely even going to talk about prokaryotic cells later. So all the stuff I'm going to teach you later about like photosynthesis and respiration and cellular reproduction, all that stuff, it's all going to be based on eukaryotic cells. So as we're going through that stuff, you could think, all right, well, how do prokaryotes work? You don't need to know it. I'm not going to teach it, but you can look it up for independent work. Anyway, those are prokaryotes. Really quickly, let's talk about eukaryotic cells or eukaryotes. They have organelles, right? So like we said earlier, prokaryotes don't have organelles. Eukaryotic cells do have organelles. The textbook definition of organelles are membrane-bound compartments or sacs with specialized functions. And what that means by membrane-bound is like I said earlier, all cells have that membrane, right? They all have a cell membrane, but also some of these organelles, they have their own membrane. So it's like a membrane within a membrane. So again, independent work idea. Here is your book definition of what an organelle is. Look it up and find another source and just compare and contrast it to. According to the book, the definition of an organelle is a membrane-bound compartment or sacs with specialized functions. And then look it up. According to this other source, this is the definition. And then just write a couple sentences how they're the same, how they're different. You can even offer an opinion if you want. I like this one better. Anyway, the size. Let's compare the size difference between prokaryotic and eukaryotic cells. So remember, prokaryotes are very simple, right? They don't have those membranes. Excuse me. They don't have membrane-bound organelles. So because of that, you can hopefully think that maybe common sense-wise, they're also going to be smaller because they're less complex, right? So prokaryotes are 0.1 to 5 micrometers wide, and eukaryotic cells are usually 10 to 100 micrometers. So you can see it's a huge difference. Again, these are things you're not going to need to know. Definitely not going to need to know their exact size. Maybe on a quiz or something, you might get a question like, what's bigger, prokaryotic cell or eukaryotic cell? The answer is eukaryotic cell. Remember, we are eukaryotes. So if you can remember that we are bigger than bacteria, I mean, granted, we're not one big cell, but hopefully that visualization will help you remember that eukaryotes are bigger than prokaryotes. Whew, any questions about that? Okay, the next thing we're going to talk about after the announcements and reminders is eukaryotic cells. We're going to dig deeper. So we compared and contrasted the two on the surface. Next thing we're going to do is really dive into eukaryotic cells a little bit more in depth. But first, the announcements and reminders. And again, I'm doing this now because at any given morning when we start a class, either half the class is not here, because they're coming in late, or they're not doing it at all because they're going to be absent. So first of all, I want to remind you this, check your emails. So many of the mistakes that are happening that I'm seeing, that I'm repeating every day when we start class, most of these things could be avoided if you check your emails. And I'll give you some examples as we go through this. First of all, I'll remind you, once again, it's a Friday, so your next week's pre-lab is due. You need to share it via Google Docs. I've been saying that from now on, I just don't care. I'm shooting for that. That would be easier for me, easier for you if you do it via Google Docs. That way I can see the mistakes, I can leave comments on your mistakes and say, hey, reply to this, and with the correct answer, so I can give you some points back. Or you can't come to lab yet because you missed this. So reply to this and let me know when you're done, which also goes back to check your emails. Because when I leave those comments, you get an email to tell me, that tells you I said those things. So I prefer you to keep using Google Docs so I can help you and you can fix things easier. If you don't, by this point, if you just want to do it, if you just want to upload it to My Online, I don't care. It's just going to make things a lot harder for you and me too, but I don't care. I don't mind if it's harder for me. I'm just trying to make things work for you. So yeah, that's due today. And again, check your emails and documents. So many people I've told them, like, hey, you need to fix this before you can come to lab. And then some of them, they don't get that until like it's almost too late or it is too late. And some people just come into lab and I'm like, you can't be here. I told you in an email, I told you in the announcements or in the comments, you can't come. And they're like, oh, I don't know. So after you turn something in, check your emails, check that assignment, make sure everything's good to go, right? Because very often, because people aren't listening to these announcements in the morning, very often people are making mistakes and I'm repeating. And then I tell them, hey, you made this mistake. And then they never know because they don't check their emails. So anyway, do that. And again, said this probably since Monday. I know I at least said it Monday before lecture, Wednesday before lecture, and then Wednesday before the lab. So I'm saying it again. This pre-lab that's due today has video questions, then it has pre-lab questions, and then it has 4 sets of hypotheses questions. So I've repeated this a lot. If you don't get it, you can't come to lab next week. It's pretty simple. If you have questions, let me know. Before you turn it in, see if I'm online, and then say, hey, I'm about to submit this. Can you look it over really quickly? And you tell me I'm in a hurry. So I can just look it over and be like, yeah, you can turn that in, right? And get credit for it. If you just want to know that, right? Can I get credit or no credit? Then I can tell you that. Or you could be like, hey, can you tell me what I missed? I can go into more detail if you want. But if you just want to say, look at this and tell me if I would get credit for it before I turn it in, please let me do that. So again, pre-lab due today. Video questions, followed by pre-lab questions, followed by 4 sets of hypotheses. Lab 3 is also due today. As you've noticed by now, I don't really care much about the due dates. So focus as far as labs are concerned. I do care about pre-lab due dates because I want you to be prepared before you come to lab. So prioritize the pre-lab over the lab. Turn it in when you can. Again, if you need my help, let me know. I can walk you through the lab. For those of you who are here, we can go through it together, get the data. The video's already online that shows all the data and that shows us doing the experiments. One thing, and I mentioned this in lab yesterday or Wednesday, that some people have already made the mistake. You have to answer all the questions. So no matter if you did it activity 1, you still need to answer activity 2, activity 3. That's all the discussion questions and the hypothesis questions. Answer all the questions. That'll be the case throughout the semester, right? No matter which one experiment you did, you're responsible for answering all the questions, which is why I was reminding you on Wednesday. I said, hey, while you guys are waiting for your stuff, you guys should go talk to your classmates who are doing the other experiments because you're going to have to answer questions about those experiments. I was reminding you to go talk to your classmates. So yes, again, answer all the questions. That will be the case throughout the semester. And then finally, once again, and I've been saying this at least since last Friday, I guarantee you, we should take a bet. I wonder how many people don't remember this and say, oh man, and they'll tell me next week, I forgot to take the exam. And then I wonder how many people will be like 2 weeks after that, like not next week, but the week after that, I forgot to take the exam. So I'm telling you right now, exam 1 and also quiz 2 are due this Sunday. You have to have it done by this Sunday. You do it via My Online, just like you do any other quiz. You have you have exactly 50 minutes to answer about 50 questions. It's a closed book exam. Obviously, I have no way to tell if you're doing a closed book exam. So on that note, I'll remind you, if you want to take more than 50 minutes, you meet me online, and I can see you, and I can see your screen, and I can hear you, so I know you're taking a closed book exam. I don't care how long it takes you to take the exam. And then finally, to remind you, the study guides for the exams are the first and second quiz. Just go back and keep taking those quizzes. It's a good practice because they're also logistically the same. Like an exam is the same as a quiz as far as how you do it. When you go in there and you open it up and you click that, right? So again, let me know if you need help. Speaking of those study guides, if you meet me online for feedback, I can give you all the correct answers for the quizzes, assuming you don't already have them, and a 5% boost to your grade. Of course, if you have 100% already, we don't need to meet. And therefore, you wouldn't get a 5% boost because it doesn't matter, you've already maxed out. Does that make sense? Anybody have any questions about what's on here?
Said for the quizzes, can you do it for the exam too?
Which would you like meet you and get the 5% boost? Yes. Which is a good time to remind you, the first 3 exams will be used to write half of the final exam. So this part, exam 1, once you're done with it, that will be one of your study guides for the final exam. So it would behoove you to meet me and get the correct answers. Like go over exam 1 after you're done with it, and I can tell you, we can look at your incorrect answers and I can give you the correct answers. And also explain why your incorrect answer was incorrect, and so on and so forth. So yeah.
Are they gonna stay open for the rest of the?
Yes, but you might need to send me a reminder. I don't remember if it was you. Somebody sent me a reminder. Thank goodness. Somebody was trying to look at quiz 1 to study for the exam, and they didn't have access to it. I'm so glad they emailed me. I fixed it. So yes, after the exam closes, everybody should then have free access to it because it'll be closed and the grades will be in the grade book, and at that point it doesn't matter, right? You just keep taking it. It'll not affect your grade after the grades in the grade book. Yeah. Great questions. Anything else? No, let me say this too, to put your mind at ease. If the grades are bad enough as a whole, you'll probably get to retake the exam as a whole. Like everybody, what'll probably happen is, depending on how bad the grades are, it might be like your first grade plus your second grade, or your last grade. If you get to retake it, you can retake it as many times as you want. First grade, last grade, right? I'm gonna add those, and then we'll just average them, basically. I'll tell you later. We'll talk about that later. But the point is, don't be too stressed. Also, if you fail it, I'll definitely let you retake it. The only catch with that is, if you fail it and get to retake it, and the whole class doesn't get to retake it, then no matter what you get, no matter how high your retake is, your grade isn't going to be higher than the lowest D. Does that make sense? If somebody with a D couldn't retake the exam, and you could because you failed, it wouldn't be fair for your grade to be higher than theirs. Does that make sense? But anyway, most likely you will be able to retake the exam. Now, hopefully the grades will be really good, and it'll be some kind of crazy averaging situation where, like, I'm counting your first grade 4 times, I'm counting your second grade once, and that'll be the average, right? You divide that by 4. But so I guess the point is, be prepared, but don't be stressed. Contact me if you want more time to take it. I generally try not to work on weekends, but for this I will. All I need to do is meet you on Google Meets. I'll have my phone. Again, I'll see you, I'll see your screen, I'll hear you. I'm not just gonna sit there and stare at you. I'm gonna go about my business, taking care of my kids, doing some dishes or whatever. I'll look at my phone every once in a while, make sure you're not cheating, okay? Put it back in my pocket and go about my business. So let me know if I can help you. And again, with all these other things I mentioned before too, is this would eliminate most of y'all's problems, not you specifically, but class as a whole. Let me look at your stuff before you turn it in. Anyway, yes, good question.
Okay, so I screwed up the lab. I didn't do all the parts. Is there a way to, like, I know you can like redo the questions, but I don't have the information. So like, how would I? Or is there a way to finish it?
Good question. Okay, so I'm glad you asked. Yeah, because it's not due until today. Past, I should say, and I should reiterate or fix that. It's actually past due today, due on Wednesday. But if you don't have the information you need for the lab, email me and I can show you exactly what I'm about to tell you. But I recorded the pre-lab lecture. So, and that pre-lab lecture, hopefully the recording showed the board that had all the data. So you can get it that way, or you can ask your classmates. I was planning on taking a picture, but I forgot. My kids have been slowly killing me. But yes, either way, it's not due until tonight. And again, I'm not that big on lab due dates. I am pre-lab, but not lab. So email me, we'll figure it out. That not just for him, anybody. If you need help, if you need data or data, let me know. And also, speaking of which, I'll go ahead and tell you this, because again, I'm very loose on the lab due dates. If I were you, I would focus on the exam, right? If you want to, don't even worry about lab 3. I mean, it's up to you how you want to prioritize and use your time, but you know, make sure you're ready for exam 1, and then worry about turning in lab 3, because I'm not going to penalize it. Now, granted, if the whole class turns in 5 labs in the last weekend, I'm not going to have time to grade it. My point is, if you turn it in on Monday, you'll probably be okay. You know, there might be like 1 point. I might deduct like 1 point if you turn it in very late, but it's a great question. Any other questions? All right, so once again, we're talking about eukaryotic cells. Actually, let me do the next attendance word. Let me think. Okay, so the attendance word should be easy since I mentioned it and he just asked it. For the lab that you have to turn, lab 3 that we did this week, that's technically past due today. Which questions do you need to answer on that lab? All the questions? Just the questions for the experiment that you did. That's the next word for the next statement for attendance. Anyway, back to eukaryotic cells. The learning objectives here are you should be able to describe the structure of plant and animal cells. Again, we're going to talk about that in a lot of detail a little bit later. So for now, it's just an introduction. You should be able to state the role of the plasma membrane. We'll have a whole portion of the next chapter devoted to that. And at that point, it'll be important. You should be able to summarize the functions of major cell organelles. The most major of them we're going to learn about in the next chapters. And that's the ones you're going to need to know about when you learn about them then. Today is just an introduction. And you should be able to describe the cytoskeleton and extracellular matrix. Again, as far as exams are concerned, not important. As far as understanding what I'm about to teach you later in the semester, not important. This is all just an introduction. Here's a picture from your lab, from your textbook. You can look at it. It's got all the parts pointed out. First of all, I've already said I'm not going to ask you many questions about this, and this is just an introduction. But second of all, I'll tell you there will be 0 questions where there's a picture like this, and I point to something and tell me, and I tell you, or ask you, what is this part? What is that part? Okay, I'm not going to do that. Then again, that would be a great extra credit question. So, all right, maybe I'll throw that in as an extra credit question, but I'm not going to ask you to identify parts. Anyway, moving forward, and I'm probably not going to stop take many questions from you because again, I just want to rush through this so we can move on to a more important chapter next week. The plasma membrane, again, like I mentioned earlier, it's made of a phospholipid bilayer that is embedded with proteins. So the phospholipids, we already talked about that on last time in the middle portion of the biological molecules section. We said a phospholipid, it's got a phosphate group, and it has 2 fatty acids attached to it. That'll be a little bit important later, but for now, you don't necessarily need to know that. 2 fatty acid chains. Oh yeah, also a glycerol backbone, even less important as far as we're concerned. But the reason I call it a phospholipid bilayer is like here is 1 layer of phospholipids. So you've got the phosphate group up top and the fatty acids sticking down. That's 1 layer. Here you have a second layer. So there you have the phospholipids within the fatty acids pointing up, right? So the fatty acids are kind of coming in on each other. That's why it's called a phospholipid bilayer. And again, no questions about this on the exam, but to understand how cells work and understand some of the stuff I'm going to teach you later, this will help you. This is a good introduction. Obviously, since it's the outermost part of the cell, depending on the cell, we'll talk about that later, it regulates what comes in and out, right? It's kind of like the border wall of a cell. Actually, I shouldn't use the word wall because we'll talk about that later too. But yeah, it's the thing that regulates what comes in and out. Some things can just come in and out with no problems, as you kind of learn when you do the pre-lab that's due today. A lot of next week's lab has to do with this. And so some things can just come in and out willy-nilly because of their size and or their chemistry. some things need some help and have to go through some of these proteins. But again, we'll talk about that later in the semester. See what that noise was, make sure we're still recording. Bear with me, please. All right, seems to be that we're still recording. So if you're missing the words of attendance, the point of it is you have to be present to get the words of attendance and be paying attention. So not to be rude, but if you missed a word of attendance, you just have to miss the word of attendance. That's kind of the point of the attendance words, right? To make sure that you were there at that time and that you were paying attention. The words themselves aren't important, right? It's just you need to, this is my proof that you're here, logged in, and paying attention. But I'm glad you asked. Anyway, moving forward. That's a phospholipid bilayer. Remember, that's what's on the outside. Hopefully you remember on the inside we have a cytoplasm. Let's talk about that very quickly. This is the contents between the membrane and the nuclear envelope. That's another way of saying it. Didn't say that earlier. Earlier I just said the cytoplasm was like a jelly-like substance where all the stuff is. So it's kind of saying the same thing. But now we're getting a little bit more specific because we're talking about eukaryotic cells. So now we also have a nucleus, which a prokaryotic cell does not. So in between that nucleus, I'll just draw it here. You've got the cell membrane. Oops. Back it up. Excuse me. And there we go. Got the cell membrane on the outside. It's the whole drawing. The nucleus on the inside. So the cytoplasm is everything in between there. It's made up of organelles suspended in a gel-like or jelly-like cytosol, which is, again, that's just the stuff, the goo, if you want to call it that. It also consists of cytoskeletons, which we're going to talk about here in a second, and also various chemicals. Again, as far as the exam is concerned, completely unimportant. As far as understanding everything I'm going to teach you later in the semester, also very unimportant. But you can look into it if you want to for independent work. Again, we're going to talk a little bit more about cytosol here in a second. But after that, if you still have any questions about it, look it up for independent work. It's about 70 to 80% water, but despite that, it still has a semi-solid consistency. What do I mean by semi-solid? Again, good grief, initiative. Gel-like, that's what I mean by semi-solid. Like a jelly, like a goo. And a lot of metabolic reactions take place here. So there will be some questions later where when we talk about these in other chapters, I'll say something like, hey, where does this reaction happen? And you're like, oh yeah, that happens in the cytoplasm. At that point, you'll need to know about it. From now, you don't need to know about it. Any questions about that slide? All right, so again, cytosol has the cytoskeleton and various chemicals. So let's talk very quickly about the cytoskeleton. You can look up the various chemicals for independent work if you want. Cytoskeleton is a network of protein fibers that helps maintain the shape of the cell. It secures certain organelles in specific positions. So some organelles kind of float around willy-nilly. Some of them need to be at specific places. The cytoskeleton helps those that need to be in certain places or that need to move from here to there, which is that bullet point. And it also enables unicellular organisms to move independently. So when you think about like a sperm with its flagella, that the flagella itself is not a part of the cytoskeleton, but it couldn't do that without the cytoskeleton. Without the cytoskeleton. So again, for the exam, completely unimportant. For the what's later in the semester, this is just an introduction. There's 3 types of cytoskeletons. Very unimportant, but we're going to talk about it very quickly. You have microfilaments that are also known as actin filaments. You have intermediate filaments, and then finally you have microtubules. And again, we're about to talk about each one of these individually. So let's jump into it. First one we'll talk about is microfilaments. These are also, again, known as the actin filaments. These are the thinnest of the 3 types of cytoskeletons. These are the things that actually move cellular components. So not just organelles, but other stuff. When things need to get moved around, this is what does it. Or this is part of the process, part of the infrastructure that allows this to happen. It also maintains the structure of the microvilli, which you don't need to know what that is, but it's right there, it is in parentheses. A microvilli is a folded cell membrane. We'll talk more about microvilli later, but even then you're still not going to need to memorize the name of it. And they're very common in muscle cells because they are responsible for muscle cell contraction. So for those of you who might be interested, like independent work, writing about muscle development or something like that, this might be a slight interest to you. So yeah, that's microfilaments, the smallest. Next, as you might imagine, is the next largest, intermediate filaments. They are an intermediate diameter, as in they're not as small as microfilaments, but they're not as big as microtubules. They help maintain the cell shape. They also help anchor organelles. And some of them are made up of keratin. So really quickly, again, not that this matters, but just so you know, to back it up, we said that these microfilaments are, I didn't say what they're made of, did I? Thinnest, really? Doesn't matter, okay. Anyway, yeah, that's what they're made of. Keratin, same kind of proteins that your fingernails are made up of. Any questions about intermediate filaments? Really quickly, microtubules. These are the thickest, right? Notice we're going from the smallest to largest. These are actually hollow tubes, right? The rest of them are not. These are hollow. They rapidly dissolve and reform. They also help guide organelle movement. And more importantly, and why these will be important later in the semester, we're going to talk about microtubules again, because these things move chromosomes around. So there will be a whole chapter where we talk about moving chromosomes around. And the way they get moved around is these things called microtubules. And then, of course, the things that I mentioned earlier, like if the sperm has that tail, that's called a flagella. And then there's also a thing called cilia, which are the little tiny hairs, right? The cells have a bunch of little tiny ones that helps them move around. And those are all microtubules. But, so you might be wondering, what's the difference between a flagella and a cilia? And then here's the difference, right? You can look at it, study it if you want, but I'm not going to ask you any questions about it. You can compare and contrast using this PowerPoint if you download the PowerPoint. Any questions about that slide? Okay, let's talk about the endomembrane system. So we're not talking about that membrane on the outside. Now we're talking about the endomembrane system. So first of all, membranes and organelles work together to modify, package, and transport lipids and proteins. So all those biological molecules we talked about in the last chapter, they need to be created. They need to be broken down sometimes. They need to be changed. They need to be moved around. And that's what this is. That's what the endomembrane system does. The membranes and the organelles work together to do these things. They include the nuclear envelope, which we'll talk about in a later chapter, lysosomes, which we're going to quickly talk about this chapter, vesicles, quickly talk about this chapter, the ER, the endoplasmic reticulum, we'll talk about that later in the semester. We'll just introduce you to it today. Same with the Golgi apparatus. Slightly important later, not important today. It is just an introduction. So the endomembrane system, excuse me, we're already talking about the endomembrane system. The nucleus, which is part of the endomembrane system. This is the most prominent organelle. This is the part that houses the cell's DNA in the form of chromatin. I should also point out there's also other DNA in the cell that we're going to talk about later in the semester. But for the most part, when we think of DNA in a cell, we should think of the nucleus. Just keep in mind there are exceptions that we'll talk about later. And this is the part that directs the synthesis of ribosomes and proteins, which might not sound that important, but I'll remind you what I said when we were talking about biological molecules, and I first introduced you to proteins. And I said one type of protein is called an enzyme. And an enzyme, there's very specific enzymes that allow chemical reactions to happen in your body. And those chemical reactions are basically everything. So when we say it directs the synthesis of proteins, and ribosomes are the things that make proteins, what we're saying is that controls your whole cell. Those proteins, usually enzymes, control all these chemical reactions. So in a way, when you think about it, that last bullet point is saying that the nucleus is control of the whole cell. And usually the way it controls it is, again, is by making these proteins, usually enzymes. So again, this will be very important later. Right now, it's just an introduction. Yeah. So the next word, answer for my next question for attendance is to answer this question. The nucleus directs the synthesis of ribosomes and what? So the nucleus directs the synthesis of two things. First of all, there's ribosomes, and there's one other thing that the nucleus directs the synthesis of. That is your next word for attendance. All right, moving on. The nuclear envelope, so we're we're not done talking about the nucleus yet, but now we're gonna talk about the nuclear envelope, obviously the part around it. It's like the cell membrane of the nucleus, if you want to think about it like that. Because it is also a double membrane. It is also a phospholipid bilayer. It is the outermost portion of the nucleus. So, right, in a sense, it is like a cell within a cell. What will be important later, but not now, this is just an introduction, is that the nuclear envelope, that membrane on the outside, it has these pores in it. And those pores let things come in and out. And later, that'll be very important, because later we're going to talk about how the DNA inside the nucleus is used to make RNA. And then that RNA leaves the nucleus, and then we use that RNA to make protein in the cytoplasm. And there'll be a whole chapter on that. So for now, again, this is just an introduction. Any questions about that slide? Ooh, 8 minutes. We're not going to finish this chapter. But like I said, that's okay. We're not going to get tested on it. I just wanted to try to knock it out because I prefer to start the next chapter on Monday. It's a good way to start things. Your brain is fresh, in theory. Oh man, it's Super Bowl Monday. I'll be in bed anyway. Look, if you want to party and have a good time at the Super Bowl, don't worry about this class. Just seriously, skip it, watch the video. Don't. I'd rather you watch the video later than come in hungover and not hear what I have to say. If you're just sitting there dazed out because you're tired, then just don't come. I don't care. Watch the video later. Send me the words, then you'll be counted as present, and you'll get your points. I mean, that's obviously the case for any day, but especially the Monday after the Super Bowl. Anyway, there we go. So we're still sort of talking about the nucleus. We talked about the nucleus itself in general. Then we just got finished talking about the nuclear envelope, which is like the membrane system of the nucleus. Now we're going to talk about that chromatin. And like I said earlier, the nucleus has DNA in the form of chromatin. So here we go. That's what it is. And this will be important later. Later, there's going to be a test question, like on maybe exam 3, I think, where I ask you, what is chromatin made of? And later, you're going to need to know that it's made up of DNA and protein. For now, it's just an introduction. These are the things, when we talk about chromosomes are made up of chromatin. And chromatin is made up of DNA and protein. What are chromosomes? Introduction right now. Later, you're going to need to know this. For now, it's just an introduction. That is the hereditary material. So all that information that you got from your mom and dad, the things that make you a merger of your two parents, that's your DNA. In eukaryotes, it's linear, meaning, I'm sure you've seen the pictures of it, but the chromosomes, you usually see them drawn like this, maybe. Right? So that's what we mean by the linear. They're in a line for eukaryotes. Now, for a prokaryote, now that you need to know it, that they'd be a circle. So their chromosomes are circular, ours are linear. And every species has a specific number. For example, later in the semester, you need to know that we have 23 humans have 23 pairs of chromosomes. That's the only one we're talking about. For now, if you want to, you can even start looking into it. Pick a species and look it up for independent work. I was wondering how many chromosomes dogs have. I looked it up, and according to this source, they have X number of chromosomes. Not that that's important, but it's interesting. I think slightly interesting. Any questions about that? All right, this part is really unimportant. The nucleolus, that is the dark area inside of a nucleus, and this is where ribosomes are made. We're going to talk about ribosomes. Ribosomes are going to be very important in conversations we have later in the semester. What's not going to be very important in that discussion is where they were made or how they were made. At that point, I'll remind you what I'm saying right now, which is you can look that up for independent work if you want. So later, like I said, we're going to talk about how proteins are made, and ribosomes have a lot to do with that. But we never talk about how ribosomes are made. So you can look that up for independent work. And I need to reread your textbook. I don't remember if your textbook mentions it and I ignore your textbook, or if it doesn't mention it either. But anyway, that's the nucleolus. So now we're working our way out, right? So then we have this thing called the ER, the endoplasmic reticulum. This is the part that's outside of the nuclear envelope. It kind of surrounds the nucleus. It's a set of interconnected membranes. Well, it's interconnected membranes. That's a typo. This is the part that modifies proteins. Again, we'll talk about that later. You have the rough endoplasmic reticulum, which is the ER. The rough ER, and then the smooth ER. The rough ER is rough because it has these little red dots on here. Those are called ribosomes. Those are the things we talked about earlier. Those are the things that make the proteins. Then you have the smooth ER, which we're not going to talk about again, but those things make the lipids. So remember that we talked about lipids in the last chapter? We didn't talk about how they were made. Well, that's how they're made. Or that's what does it. Now, again, this is the last time we're probably even going to talk about smooth ER. So for independent work, you can look that up. How are lipids made? How does the smooth ER make lipids? Next, we have the Golgi apparatus, also sometimes called the Golgi body. This is a series of flattened membranous sacs. They sort, package, and tag proteins and lipids. So again, think about like starting from the inside, working out is what we're doing, right? Because we started talking about the nucleus, we're working our way out. And a lot of the time, we don't really focus on lipids. But again, this whole protein thing will be important later. So we're going to make these proteins on the rough ER with the ribosomes. Then they need to be moved and packaged and tagged. And you'll see what I mean later. Actually, we won't even talk about that again, I don't think. But when I say tag proteins, what I mean is that your cell will make a protein, and then it'll put some sort of molecule on it. So then your cell knows where that thing needs to go. So the Golgi apparatus is almost like the post office in a way, or the shipping center of a factory. The factory is making these things, and once they're ready to go, they go to the Golgi apparatus, and where they tag and package it and make it ready to go. Or send it out, I should say. Obviously, the receiving side of this is going to face the ER, the endotransmic reticulum, because that's where the proteins are coming from. And then the other side is where it releases the lipids and the proteins. So that should make sense. But again, we're not going to talk about the Golgi apparatus again in this semester. So there you go. There's your introduction to the Golgi apparatus. Now we'll say goodbye to never learn about it again. All right, now we're kind of done. We're done talking about this whole, we're done working our way from the inside of the nucleus on the way out. Now we have other things called lysosomes. These are your garbage disposals. These things use digestive enzymes to break down stuff. So if there's something in your cell that doesn't need to be in there, either because it doesn't need to be there anymore or should have been there in the first place, lysosomes take care of that. They break things down. So what you'll have is these big macromolecules. And as you remember, macromolecules are made up of those are polymers made up of monomers. Well, that breaks it down so we can get those monomers, and then your cells can use those monomers to build something new. And again, like I said, not only do they destroy things that should be there, but we don't need them anymore, but they also destroy things that shouldn't have been there in the first place, which is that last bullet point right there. They destroy disease-causing organisms. All right, let's see what we can get before 8:50. Then we have vesicles and vacuoles. These are also membrane-bound sacs. They are responsible for storage and transport. Vacuoles are larger than vesicles. Not that I'm going to ask you any of this, and both of which can break down macromolecules. And they do this after they break them down. They fuse with the cellular membrane, and they kind of like get it out, right? So we broke this thing down. We don't need it anymore. Fuse with the cellular membrane and just get rid of it out of the cell. There'll be a term for that you're going to learn in a later chapter. Ribosomes, I'm not even going to talk about this now because we're going to talk about I've already talked about them a little. This will be another chapter where you definitely need to know about them. Same with mitochondria, and that's what we'll end the word in today's lecture. And the last word for attendance is not a word at all. Just send me a question about anything that I talked about today, including not just the chapter, but including the announcements and reminders. If you have a question about how labs work, please ask me. Let's get things cleared up. And that's it. I'm going to be going to my office. I'll be on mine at least until noon if you need me, or in my office. Don't forget, the exam is due Sunday. I'm guessing